Post-viremic COVID, vitamin D receptor and toll-like receptor therapeutics for cytotoxic and inflammatory COVID injuries. These are notes for the COVID Zoom group meeting for July 10th. Key points. Kidney injury is common in progressive COVID patients. It is estimated that approximately 70% of progressive COVID patients experience some form of acute kidney injury. This injury has been defined as an acute Fanconi syndrome, which involves damage to the proximal convoluted tubules of the nephron. And this often precedes acute kidney injury, which is a more progressive form of the injury which is observed in hospitalized COVID patients. Point 3, CoV-2 spike protein, the S1 fragment, amplifies TLR-mediated immune responses to the bacterial toxin LPS, lipopolysaccharide, leading to and contributing to hyperinflammation. In today's notes, we will look at the integrated physiology at the cellular level involving the interplay between the vitamin D pathway involving calcitriol binding to the VDR receptor and the interaction of this pathway with toll-like receptors, specifically TLR2, TLR3, and TLR4. We'll also examine how antimicrobial peptides, AMPs, contribute to the innate immune defense system, which is essential for withstanding COVID infections prior to the involvement of antibodies being generated by the adaptive immune system. Point 4, calcidiol 25D and calcitriol 125D have been found to provide substantial COVID morbidity and COVID mortality benefits. These results come from 2 groups in Barcelona, Spain. Point 5, FGF23, a stress and injury hormone, suppresses the synthesis of calcidiol 25D and calcitriol 125D by upregulating 24-hydroxylase and downregulating 1-alpha hydroxylase. These pathways have been known for decades in renal tubule cells. However, these same pathways are involved in the regulation of calcitriol synthesis in immune cells, as well as diverse types of endothelial cells and epithelial cells in general, including the cornea. So it is important to look at COVID histopathogenesis literature to help address and resolve current COVID treatment controversies. In today's notes, we will examine how vitamin D pathway therapeutics and toll-like receptor therapeutics can be potentially repurposed to treat long COVID. Long COVID can include COVID-induced kidney injuries and their associated endocrine deficits that will occur because of injury to both the nephrons in the kidney as well as the interstitial cells, which secrete important hematopoietic hormones such as erythropoietin. It is the deficit of calcitriol which is of immediate importance because calcitriol is critically important for the activation and maintenance of the innate and adaptive immune responses to the COVID infection. This paper discusses the statistics of recovery rates in new onset kidney disease in COVID patients which have been discharged from the hospital. This means these patients had no existing kidney disease prior to COVID and their hospitalization. On average, it takes approximately 4 months to fully recover from the acute kidney injury, and I should say acute Fanconi syndrome injury, which occurred as a result of COVID. Acute Fanconi syndrome precedes acute kidney injury. So additional terms to describe the acute Fanconi syndrome is an acquired Fanconi syndrome or an incomplete Fanconi syndrome. It involves injury to the proximal convoluted tubule cells and results in proximal convoluted tubule dysfunction, particularly the loss or depression of calcitriol synthesis. Key words, it is important to recognize that this is a physiological and endocrine dysregulation, which is occurring as a consequence of both the COVID infection, cytokine storms, as well as therapeutic agents such as steroids, which are applied to control the hyperinflammation associated with COVID. Damage to the microvilli, the brush border of the convoluted epithelial cells, is a long-term histopathologic deficit which impacts severely the genesis of calcitriol in the serum, because the kidney proximal convoluted tubules are a primary source for the circulating calcitriol. So the loss of brush border during COVID is something that will persist in long COVID patients, and this must be recognized by practitioners and patients and helps motivate the discussion of using calcidiol and calcitriol as therapeutic interventions for long COVID, post-viremic COVID patients. In regards to the proximal convoluted tubule cells, the epithelial cells, which have microvilli on their apical surface, this is the locus of vitamin D receptors, which bind circulating 25D and also circulating 125D in the urinary filtrate. This is the fluid coming through the glomerulus. These vitamin D metabolites are captured, brought to mitochondria, where they can be modified by hydroxylases, and then through a vitamin D binding protein carried to the nucleus to evoke changes in numerous types of gene expressions. This diagram illustrates the importance of two hydroxylases. Now, these are localized in the inner membrane of mitochondria, not illustrated here, but the upregulation of 24-hydroxylase results in a metabolic shunt that moves 25D and 125D calcitriol to inactive forms. At the same time, 1-alpha hydroxylase, when it is downregulated, the cell is unable to convert. Use the term conversion in this endocrinology literature. The conversion of 25D to 125D is impaired, a second blow to the synthesis of calcitriol. A third and very important deficit that needs to be stressed for long COVID is the loss of the microvilli on the brush border of the epithelial cells. Without this structural element, the vitamin D receptors cannot be localized in the appropriate location to retrieve these vitamin D metabolites from the urinary filtrate. Thus, a structural deficit leading to an endocrine deficit, thus leading to immunodeficits. In previous YouTubes, we have discussed this pathway very well illustrated, showing the various failure points in the vitamin D biosynthesis pathway. Again, the term vitamin D is very ambiguous, so it's important to distinguish the pro-hormones from the active hormone 1,25-D calcitriol. It has been known for over a century that lack of sunlight results in rickets, a failure mode at the liver inflammation, fatty liver disease, hepatitis, results in the block or the metabolic shunt of conversion of vitamin D3 into 25D. This same failure mode exists in the kidney, inflammation of the kidney, which occurs with chronic kidney disease. A circulating hormone, FGF23, produces these changes in hydroxylases. Hydroxylase dysregulation impairs mitochondria and the endoplasmic reticulum in converting these metabolites into calcitriol. The resulting calcitriol deficit can lead to what is known as vitamin D resistance, which is common in autoimmune disorders. So one failure mode is the inability of the body, the kidney or immune cells or epithelia, to synthesize calcitriol. Another is the downregulation of vitamin D receptors in cell membranes. Cells can also be impacted by the chronic use of steroids, and in the case of COVID infections, the use of high-dose steroids impacts the expression of the hydroxylases, 24-hydroxylase and 1-alpha-hydroxylase. So, in terms of COVID, the disease itself and the production of hormones such as FGF23 and the application of steroids can produce calcitriol and calcidiol deficits. This is important because of two salient papers that have been published this year. First, a retrospective study showing that calcitriol 125D supplementation in chronic kidney disease patients, these were patients that were taking calcitriol prior to acquiring COVID. The authors of this study determined that calcitriol at 0.4 micrograms per day provides a major survival benefit as well as a morbidity benefit compared to chronic kidney disease patients that were maintained on lower amounts of calcitriol, 0.1 microgram per day. This emphasizes that calcitriol is a rescuing, implies that it's a rescuing agent, and that a depletion of calcitriol may be a contributing factor to immunodeficits, leading to increased inflammation, immunodeficits that could result in bacterial and fungal infections. So there's many potential benefits of calcitriol that need to be examined, prompted by the results of this paper. Now, the next section of this discussion concentrates on the newer paper, also from our group in Barcelona, calcidiol, also known as calcifediol. It too provides substantial morbidity and mortality benefits, but in this case applied to hospitalized COVID patients. The prior study, the vitamin D metabolite was applied prior to the infection. In this more recently published study, calcifediol, 25D, is applied early on during hospitalization. So this brings up an important key public health issue. How do practitioners and members of the public view vitamin D supplementation. Oftentimes, patients and practitioners are advised to prescribe a certain amount of vitamin D per day, 1,000 international units per day, 4,000 international units per day. These, unfortunately, do not work well for the acute, highly inflammatory states of COVID for the following reason. The inflammatory states, as well as the application of drugs like steroids, dramatically alter hydroxylase expression. The guidelines that were used for bone health, for the elimination of rickets, do not apply to the benefits of higher levels of calcidiol and calcitriol, which they provide for the immune responses that are needed to combat COVID. Now, this is a major source of controversy that is present through many of the current discussions. What is the appropriate target for calcidiol levels in the serum? Now, this is not the endpoint. As 25D is inactive. It merely means that this level of substrate is a good target to help withstand the impacts of COVID. It does not mean that calcidiol is the therapeutic metabolite. That is calcitriol, and it must be able to be converted from 25D to 125D for this to occur. So in the case of a progressive COVID infection with COVID lung, ground glass opacities, strong evidence that these are associated with, in many cases, bacterial and fungal growth taking place in tandem with the hyperinflamed regions of the lung. The ground glass opacities correspond to hyaluronic acid hydrogels, and in this hypoxic immunodeficient environment, this is a place where secondary opportunistic infections become being established. So serum calcitriol levels in healthy individuals range from around 20 or 30 to 60 picograms per milliliter. Different units are used to describe the serum calcidiol levels. Different countries argue for different amounts of calcidiol as a target, but range from 30 to 100 nanograms per mil is now being discussed as a healthy target zone. So moving patients to a higher level of calcidiol, 25D, is what the group in Barcelona has now published on. There were substantial benefits, morbidity and mortality benefits, by moving calcidiol levels, calcidiol is a term for it, quickly through oral supplementation. And I encourage viewers of this YouTube to pause and take a look at this paper in detail. This brings us to section F of this discussion. Vitamin D metabolites, antimicrobial peptides, toll-like receptors, and COVID. How to build context for these therapeutics, deriving these protocols from immune response combinatorics. So this is a very nicely constructed, compact immunoendocrine diagram. However, even though it shows topologically how manipulating one factor such as 25D or 125D calcitriol provides benefit to stimulating the expression of antimicrobial peptides like human defensin beta 2 or the calcilethodin molecule LL37, how these are important to withstand bacterial and fungal infections. These these diagrams do not illustrate kinetics or kinematics. And I mean pharmacokinetics of the metabolites and changes, and the kinematics of how these metabolites are moving through the body, short range through diffusion, long range by hydraulic forces moving bodily fluids. In order to understand how effective any of these metabolites are, one has to utilize Gray's anatomy and multiple levels of integrated physiology. So we've talked about this in prior discussions, failure modes for vitamin D hydroxylation. A major site of failure is in the liver, another one is in the kidney. And both of these could be likely to occur in COVID patients because of hyperinflammation impacting these visceral organs. So the upregulation of 24-hydroxylase, the downregulation of 1-alpha hydroxylase would result in a metabolic shunt appearing in the liver, preventing the conversion of cholecalciferol, vitamin D3, into calcidiol. Now, placing that in context, the doctors from Barcelona who recently published on calcidiol 25D, providing survival and morbidity benefits for COVID patients, are supplementing past this metabolic block, this first failure mode. And by doing this in early stage COVID patients, they are supplying the substrate for the kidney, the immune cells, and various epithelial cells to convert 25D into 125D, calcitriol. Now, as the disease progresses and proteinuria is observed and steroids are applied, it is very likely that the kidney's ability to form calcitriol will be compromised. For that reason, it's important to look at the earlier paper from a different group in Barcelona, how calcitriol was able to provide survival benefit for patients with existing chronic kidney disease. This illustrates how important calcitriol is as a rescuing agent for probably multiple aspects of the endocrine and immune responses. So in the future, practitioners need to discuss whether there are changing ratios that will be needed to treat COVID during the viremic period and the post-viremic period. How much calcidiol, how much calcitriol should be given to patients to help support them through these time windows. There are numerous reasons why calcidiol and calcitriol should be applied, because calcitriol is the key metabolite to activate numerous vitamin D responsive genes in the genome. Approximately 2,000 genes are vitamin D responsive out of 20,000 genes in the human genome. And these vitamin D responsive genes provide multiple benefits, multiple pathways, such as the induction of antimicrobial peptides to suppress numerous types of viruses, blocking viral entry and replication, induction of autophagy, which is impaired in COVID infections. Also, suppressing TLRs, not completely, but modulating them. I would prefer to use the word stabilizing the TLR responses. And today we will bring into the discussion the need to integrate the thinking about the calcitriol, vitamin D receptor signaling pathways, and the toll-like receptors, which is so well illustrated in this summary diagram. So the toll-like receptors impact the canonical pathway of inflammation, a nuclear factor kappa beta transcription factor, which is upstream in the production of many pro-inflammatory cytokines. So the modulation, the activation, stabilization, or modulation, or inhibition of toll-like receptors has to be a key point of discussion for COVID infections and post-viremic COVID patients. So moving forward in this discussion, let's first look at toll-like receptor 4. This paper asked the question, is it involved in cardiometabolic comorbidities? Of inflammation may be the core concept that needs to be discussed in COVID, whether it's the heart, the lungs, the kidney, the pancreas. Perhaps in the year 2020, too much emphasis was focused on specific organs as opposed to a view of a more systemic impact of circulating metabolites like the COVID spike protein fragment S1, FGF23. So to be cellular-centric or tissue-centric or organ-centric, that's important, but it's also important to integrate them into a broader framework, especially when looking at the role of these toll-like receptors, which are found in all cell types, including platelets. The spike protein can interact directly with the toll-like receptor. This is very disturbing and potentially informative about how the spike protein fragments, the S1 fragments, are metabotropically toxic, cytotoxic, by producing a direct stimulation of TLR4. Some highlights from this paper. Some inflammatory diseases are related to severe forms of COVID-19, characterized by immune system overactivation. Now, this is a very insightful comment. I'd like to add that the analysis of poisonous snake bites, particularly vipers, may be an important analogy to what is being seen in a hyperinflamed, overactivated immune system. Point 2, the COVID-2 spike protein combined with the TLR4, leading to the production of pro-inflammatory cytokines. Inflammatory diseases are risk factors for COVID: hypertension, diabetes, obesity, atherosclerosis. All of these are connected to continued activation of toll-like receptors. And the toll-like receptor 4 signaling pathway is connected to these inflammatory diseases, and perhaps because of this convergence of TLR on TLR4 activation, which drives nuclear factor kappa beta, that COVID is exploiting a defense system and exaggerating the impact on many visceral organs simultaneously by activating TLR, motivating the desire to attenuate or stabilize this pathway. And one of the key non-toxic inhibitors which is available is low-dose naltrexone. And in prior presentations, we have discussed the importance of antimicrobial peptides, AMPs, to withstand COVID eye infections and nasal infections. I utilize this slide again to emphasize the hypothesis by Suzuki that spike protein fragments, the S1 fragment, are the mobile, highly pathogenic or pleiotropic toxins that can be bound to albumin, transcytosed across epithelia, enter the CNS, accumulated into the serum. How several types of AMPs can bind to the spike proteins and neutralize them. Why is this important? As the more contagious variants, I should say, not only contagious, but more pathogenic variants have arisen. It is important to recognize that the evolution at the nanoscale, particularly at the receptor binding domain, is altering the kinetics and the kinematics of the infection. Not only does it allow the virion to bind more effectively to the ACE2 receptor, it's now known that the spike protein can bind to toll-like receptors, and they may be utilizing toll-like receptors as a means of entering cells. So this brings up a very important, broader issue of the role of toll-like receptors in various stages of COVID infections and COVID recovery. For instance, the antimicrobial peptide LL37, the only cathelicidin gene product in the human genome, which is essential for antifungal activity, it also plays antibacterial functions and also neutralizes viruses. So LL37 is essential for the innate immune response. However, LL37 has important roles of activating toll-like receptors, which could be compromised by COVID infections. Let's look at the role of LL37 in the activation of toll-like receptor 3 and its role inside endosomes. Now, endosomal physiology, the nanophysiology of endosomes, is incredibly important for understanding the hyperinflammation of COVID. So here we see two toll-like receptors, TLR4, which is described as a membrane-associated toll-like receptor, although it is internalized into endosomes, versus toll-like receptor 3, which is described as a, quote, endosomal toll-like receptor. So LL37, this antimicrobial peptide, its natural ligand is double-stranded RNA, which is indicative of a viral infection. Mammalian RNAs are single-stranded, so the presence of double-stranded RNA is an important signal for cells of an ongoing viral infection. So when this ligand binds to the receptor, and I'm talking about LL37 as the, quote, receptor for this ligand, the complex then interacts with FPRL1, and that is brought into an early endosome, combined with an endosome containing TLR3. In the acidification that takes place in the early endosome, there's a dissociation of the double-stranded RNA from the surface receptor and from the LL37. And now the double-stranded RNA can interact with toll-like receptor 3, which sets off the canonical pro-inflammatory pathway, which pivots around nuclear factor kappa beta expression. And this transcription factor is upstream of numerous cytokines. So the importance of this second messenger pathway, or the signal transduction pathway, is that toll-like receptor 3, as a detector for the viral infection, is being activated, utilizing LL37 as an extracellular scavenger or detector for this ligand, bringing it to the cell, allowing it to interact in endosomal compartments to perform the activation of this toll-like receptor 3. So this is a model by which LL37 enhances the activation of the inflammatory response to this double-stranded RNA. Now, double-stranded RNA, single-stranded RNA will be released. This is viral RNA from dead or dying cells that have been infected with COVID. So besides the S1 spike protein fragment, circulating double-stranded RNA from COVID is certainly one of the pro-inflammatory signals that's setting off and contributing to inflammatory states throughout the body. So there's 10 toll-like receptors in the human genome, several in the cell membrane, others in endosomal compartments. How pathogenic molecules are perceived, like lipopolysaccharide, double-stranded messenger or double-stranded viral RNA, the spike protein and the spike protein fragment. All of these pathogenic agents will be interpreted by different members of the toll-like receptor family, and they feed into a broader set of pathways that are both pro-inflammatory and anti-inflammatory, which are severely imbalanced in COVID patients because of the inactivation of ACE2 receptors, which provide major, quote, anti-inflammatory tone to the immune system. So this paper, I encourage viewers of this YouTube to read this paper as a means of viewing the layers of structure within the inflammatory response. Because in terms of treating long COVID patients, residual inflammation, there are molecular players like spike proteins, fragments, double-stranded RNA, pro-inflammatory cytokines that can persist and factors released from damaged cells, setting off toll-like receptors that will persist in producing these localized and systemic inflammatory responses. This paper also describes how just a single endotoxin, LPS, by interacting with toll-like receptor 4, combines and augments inflammatory reactions that are promoted by pro-inflammatory cytokines like tumor necrosis factor, complement injury, the impact of these pro-inflammatory cytokine cascades produce microvascular coagulation, which produces ischemic reperfusion injury, which can occur in any organ. So the continued imbalance and activation of these pathways is what is particularly damaging in progressive COVID patients and results in the prolonged recovery period that some COVID patients experience. And this gets into a little more of the molecular details of the interactions of LPS with the toll-like receptor 4 pathway. Now, one question is, why is COVID so pro-inflammatory? Why is there such high degrees of hyperinflammation? It's important to recognize that this was first proposed by a French doctor in early stages of COVID, early 2020, that bacterial co-infections are occurring in the lungs of many COVID patients. And with gram-negative bacteria proliferating in the lung, the release of lipopolysaccharide, LPS, and the activation of toll-like receptor 4 activates an innate immune response, an inflammatory response. It's now been determined that the spike protein, the CoV-2 spike protein, can bind to LPS, and that complex hyperactivates the toll-like receptor 4 pathway, and it boosts the inflammatory activity from these stimulated cells. So here, the molecular ecology of the extracellular environment is dramatically altered because of the presence of the spike protein and the S1 spike protein fragment binding to LPS, forming a super agonist for the toll-like receptor pathway. Again, activating the canonical NF-kappa beta pathway. Now, in this study, numerous experiments were done, but this one is particularly instructive. They used a transgenic mouse line that had a reporter gene for the nuclear factor kappa beta being driven by that gene's promoter. They were using a luciferase gene expression, which allows one to localize the upregulation of the NF-kappa beta gene. And the conclusion they derived from this experiment is that, diagrammed here, LPS aggregates at low concentration can dissociate and then attach to the spike protein, either on a virion or the S1 fragment. And then that free lipopolysaccharide can bind with the spike protein to the toll-like receptor and activate it. At higher concentration, the LPS aggregates remain intact and can form aggregations with LPS and spike protein fragments and interact with different receptors, two different potent means of activating inflammation. Now here is the mouse luciferin bioluminescence imaging experiment. Now this is a recently developed bioluminescence imaging methodology by which reporter genes can be visualized. The long wavelength photons coming from the firefly luciferin, that's the substrate, when it is acted upon by luciferase, the luciferin is cleaved, releases a photon, and in this case, red photons are moving through the tissues, through the skin, and being imaged with a low light bioluminescence imaging camera. And through this, they can examine the response of this transgenic mouse to various injected agonists, such as lipopolysaccharide on the left, or lipopolysaccharide plus spike protein on the right. And you can see, without going into much detail, that where LPS is applied in combination with spike protein, there is a larger inflammatory reaction monitored through this indirect bioluminescence assay. Now finally, we'll shift to the discussion of FGF23, which is very likely to play a pivotal role in many stages of COVID. Even from the earliest infections, FGF23 may be produced as a response of the localized COVID infection. FGF23 is able to inhibit extrarenal synthesis of calcitriol 125D, and in this case, in human monocytes. But FGF23 is also able to inhibit calcitriol synthesis in the kidney, in various epithelia, and a number of immune cells as well. The importance of that is that without a local source of calcitriol, each of these cells, immune cells or epithelial cells, will be driven into states of hyperinflammation. And that is because the activation of vitamin D receptors helps suppress the nuclear factor kappa beta pathway. Calcitriol, through activation of VDRs, helps stabilize the toll-like receptor pathways. So FGF23 is a distress and injury hormone. Its suppression of calcitriol may be a normal, natural response to a local injury or an infection to help stimulate the immune response locally. However, when FGF23 circulates at high levels, it produces low-level or more exaggerated levels of inflammation, chronic inflammation in many tissues, and contributes to many progressions of disease states, especially in older people and people with autoimmune. So I believe that FGF23, even though it is unfortunately not reported on in many COVID papers to date, we can rely on older FGF23 literature for context. So, ironing out the crosstalk between FGF23 and inflammation. And one of the key figures up front is the impact of FGF23, in this case coming from osteocytes in the bone, impacting the regulation of hydroxylases, which we've emphasized repeatedly in this presentation. Hydroxylase dysregulation is the overall concept that helps integrate these endocrine, immunoendocrine signals, which can be associated with damage or pathogens, infections. FGF23, by modulating these hydroxylases, alters the synthesis of 1,25D calcitriol, not only in the kidney, the proximal tubular cells, but in immune cells and epithelial cells throughout the body. So this paper clearly delineates renal calcitriol synthesis from extrarenal calcitriol synthesis. In both cases, 1,25D, the central immune hormone that's needed not just for bone metabolism and bone health, but immunomodulation, anti-inflammation, and antimicrobial activity. A deficit of calcitriol has severe consequences on both the inflammatory state of tissues and organs, as well as their ability to resist microbial infections. So it's very likely that there is an evolving calcitriol crisis going on in COVID, first starting off at the local sites of COVID infection, then expanding as mobile agents like the S1 spike protein fragment, double-stranded viral RNA, and FGF23. This combination of soluble, mobile, pathogenic molecules would be having progressive and increasing impact on calcitriol synthesis in the kidney and in other sites as well. Once 24-hydroxylase is upregulated in response to these stimuli, the mitochondria in kidney, the mitochondria in immune cells, corneal cells, endothelial cells will be unable to make adequate levels of calcitriol, driving them into inflammatory states, compromising their ability to make antimicrobial peptides. I like to emphasize the importance of this paper, which comes from veterinary literature, critical care for animals. It's a very well illustrated, diagrammed synopsis with some pharmacokinetics describing the use of calcitriol, calcidiol, monitoring parathyroid hormone levels, as well as fibroblast growth factor 23 in the context of chronic kidney disease, which is common in aging domestic animals. So just briefly, some descriptions of how um, vitamin D binding proteins are able to recover vitamin D metabolites from interstitial fluid and lets the filtrate and move it, modify these metabolites, and secrete them into the interstitial fluid of the kidney where they're transported into the capillary bed and exported through the body. Description of how pro-inflammatory pathways can impact the health of the kidney, resulting in numerous renal dysfunctions, pathogenesis, resulting in proteinuria, tubular hyperplasia, monocyte infiltration, and fibrosis. And a loss of vitamin D receptors impairs calcitriol synthesis, vitamin D receptor activation. It is a downward spiral that can occur. Now back to FGF23. Here we see a diagram where inflammation and the activation of signaling pathways like nuclear factor kappa beta result in the expression of FGF23. And then a series of diagrams describing how FGF23 fits into normal metabolism, regulating calcitriol levels, helping to suppress high levels of phosphate in the serum. And this is in a healthy animal. And then in early chronic kidney disease, as well as late chronic kidney disease, how FGF23 and suppressing calcitriol synthesis produces numerous cytopathologies and histopathogenesis that results in a downward spiral in chronic kidney disease. So all of those pathways are very important in COVID because they're accelerated, very rapidly accelerated. And so to understand progressive COVID, it's important to hybridize knowledge from nephrology, endocrinology, immunology, pharmacology. By doing the systems and integrative physiology analysis, coupling that with histopathogenesis literature, we can come to understandings with certain key points emphasized. First, kidney injury is common in progressive COVID patients. Second, it begins with a condition now is called acute Fanconi syndrome, which is known from other toxic agents, which can occur from heavy metals, drugs, viral infections, and even snake bites. And that acute Fanconi syndrome precedes a more serious condition known as acute kidney injury. For long COVID patients, an assessment needs to be made to understand how much damage occurred during the course of their viremic period, how much calcitriol synthesis deficits are currently occurring in the post-viremic period, how much calcidiol and calcitriol is needed as a first-line intervention for these types of kidney injuries in the long COVID individual. Point 3, it's now known that spike protein fragment S1 amplifies the toll-like receptor-mediated immune responses to bacterial endotoxin LPS, one of the reasons that's driving hyperinflammation, and maybe one of the key reasons underlying the hyperinflammation resulting in the ground glass opacity seen radiologically in COVID lung. However, the same type of amplification needs to be considered for other places where bacteria are common, for instance, the gut, the nasal cavity, and the eye, cornea. So we discussed in this presentation the integrative physiology at the cellular level of how vitamin D receptors, VDRs, vitamin D metabolites, calcidiol 25D, calcitriol 125D, activating the VDR pathways, help modulate the behavior of the toll-like receptor pathways, the TLR pathways. The activation or the upregulation expression of antimicrobial peptides by these pathways is essential for combating various pathogens, viruses, bacteria, and fungi. Compromising calcitriol or hyperactivating the toll-like receptors imbalances the innate immune system in the most upstream elements of these cascades. For that reason, looking at calcitriol, calcidiol, and agents like low-dose naltrexone is very important to look at those as potential therapeutic agents for new types of cocktails, new formulations. Unfortunately, they are evaluated in isolation singly. In order to have maximum benefit, they need to be applied in cocktails, and that needs to be considered in the construction of future clinical trials or in the treatment of individual patients. Point 4, calcidiol 25D, calcitriol 125D have been found to provide substantial benefits for COVID morbidity and COVID mortality. And I encourage people to read these recent papers from 2 separate groups in Barcelona. Finally, point 5, FGF23, fibroblast growth factor 23, is known to suppress the synthesis of calcidiol and calcitriol in diverse cell types and tissues by 2 pathways, one upregulating 24-hydroxylase and downregulating 1-alpha-hydroxylase. The analysis of COVID and the productive application of therapeutics must include a discussion of epigenetics, transcription factors, feedback loops. This is the language of endocrinologists, rheumatologists, developmental biologists. Metabolomics is anchored to the transcriptional events taking place in the nucleus, the impact of those events on the functioning of organelles of mitochondria and endoplasmic reticulum, the dynamics taking place in endosomes, multivesicular bodies, and the elimination of pathogenic molecules in locations like lysosomes. Endomembranous compartments, the modulation of activities in organelles. This is integrated pathophysiology and recovery at the subcellular level. And it is only through a prolonged discussion and testing and rejecting of concepts that these particular therapeutics listed here, calcidiol, calcitriol, and as we've discussed, low-dose naltrexone, can be adequately applied to especially the more virulent, more pathogenic forms of the COVID variants.